I still have the photo profile picture. I have romance pictures. No. It's just you as a baby. I'm definitely contemplating on doing that. I have
Alright, so there are many metabolic pathways, and each of them has many steps. In fact, there are too many steps. So someone like you who is knowledgeable in organic chemistry, you can say, I can do that reaction in much fewer steps. Why does the cell take so many steps to do it? And then a small child can say, I can do a degradation step in one step. Just take some glucose and throw it in the flame and it will burn to give carbon dioxide. That's exactly what you're doing in the cell. When you say you're burning calories, means you're burning glucose to give carbon dioxide and water, which can be done in one step. But the cell takes 28 steps to convert one molecule of glucose to carbon dioxide and water. So why does it take so many steps? And that is the same question for all pathways. There are many steps. So what are the reasons for having multiple steps for all pathways? There are 4 reasons, of which the last 2 are more important. One is limited reaction specificity of the enzymes. We know enzymes are very specific for their substrate. Enzymes are extremely efficient. They're always 100% efficient. Everything is good about enzymes. But they can do only a limited number of steps in the reaction. All mechanisms will have only a limited number of steps. You cannot do very complex reaction with one enzyme. So if it is a complex reaction, there has to be multiple enzymes to do those different steps. So that's one reason that every step is, every enzyme is highly specific and for doing simple reaction, not complex reactions. Secondly, you can use the intermediate So if you have multiple steps, that means you have multiple intermediates. And those intermediates can serve as metabolites for other pathways. So if you have a pathway going from one molecule to another molecule in one step, there is nothing in between. But if you have multiple steps, then this thing can be part of another pathway. This thing can also come from another pathway. So the pathways can be interconnected if you have multiple steps, and that's how all pathways are. There are intermediates can be used for other pathways, and there can be other pathways that feed into one pathway. So that's another reason why you have multiple steps. The more important reasons are the last 2. One is efficient energy use. So reactions will require energy, some reactions release energy, and the whole idea of burning glucose is to make ATP. So when one glucose is burnt, to give you 6 molecules of carbon dioxide and water, I'm not balancing it. So one glucose will give you 2800 kilojoules of energy. And what will this energy be used for to make ATP? If this reaction was done in one step, then only one molecule of ATP can be made, because then that's how the enzymes work. They have limited reaction specificity, and you never make more than 1 ATP in a reaction. So all this will be used to make 1 ATP. So if this is the one step, if this happened in one step, you release this much energy, then that will give you 1 ATP, and 1 ATP is about 30 kilojoules. It takes about 30 kilojoules of energy to make 1 ATP, and what will happen to the remaining energy? All that remaining energy, 2,770 kilojoules, will be released as heat energy, which is not what you want. There will be too much of heat released, and that is not very efficient. The whole idea is to make ATPs, and so multiple steps. In fact, there are 28 steps. Then this energy is used to make about 32 ATPs. All right, so there's a contradiction here. I just said one reaction will make only 1 ATP. But how do you make 32 ATP in 28 steps? That's not something that was understood until the late 1980s. So we will come to that later in a later chapter when we do oxidative phosphorylation, that you can make more than 1 ATP. But the idea even today is that in a chemical reaction, it is a stoichiometric reaction, so one reaction will usually give you 1 ATP. So approximately, let's keep that in mind, that you do multiple steps to get multiple ATPs. Same thing with synthesis. So this is about degradation, where you're releasing energy, and you don't want all that energy to be released as heat energy, which will be actually very bad for you, because that much of heat energy released will raise the body temperature, and basically the person dies. So that's not desirable. Some reactions will always release some energy. So most of the reactions that you see in any pathway, if they are releasing energy, how much energy does it release? So most reactions will have a energy released something in the range of maybe 60. So there is no point in releasing maybe, let's say, 100 or 200 kilojoules of energy, because all you can make is 1 ATP. So if a reaction releases 200, then 170 is released as heat, which is a lot. So most of the reactions will be in the range of 30 to 40 to 60, in that range, and maybe there is one or two that releases 100. So what happens to the rest? So most reactions will have a delta G, maybe between 30 to 200. And that means they will make 1 ATP. If it is 100, then 70 kilojoules will be released as heat energy. If it is 40, then 10 kilojoules will be released as heat energy. And that is the amount of heat that you need for maintaining your body temperature. So nature has evolved so that the energy that we release from the various reactions keeps your body warm by releasing that extra energy. You don't want exactly 30 for all reactions, then no heat will be generated. So heat is generated in most of these reactions. But you don't want too much excess, you don't want 200 or 300, because that will mean too much of energy released. We also have synthesis reaction, which means you are making bonds, and always remember, whenever a bond is made, it requires energy, and that reaction must use a source of energy like ATP. So all synthetic reactions where you are forming a bond without breaking another bond, that means you are using ATP for that. And ATP provides you only 30 kilojoules of energy, actually close to 31. So if a reaction requires, let's say a reaction has delta G equal to plus 60 kilojoules, that means this is a reaction that requires 60 kilojoules of energy. Where will that energy come from? We don't have a mechanism to provide that much energy, because enzymes will work only with 1 ATP at a time, and they can get only 30 kilojoules of energy from there. and that is not enough to carry out this reaction. Which means there is no reaction in the cell, or in all these hundreds of reactions that we have, there is nothing that has a delta G, a positive delta G of more than 30, because all your energy comes from ATP, so you cannot have a delta G of 60, that reaction cannot even happen. You cannot combine two ATPs to get one reaction. So that means this kind of reaction is not there. So all of them must have delta G less than 30, because you have to get that energy from one ATP. And that again means you need multiple steps for all pathways. So as I said, the third one and the fourth one are the most important reasons why there are so many steps. So this is the efficient energy use. And secondly, all pathways are regulated. That means the pathway is turned on when you need it. When you don't need it, then the pathway is not on. So all these reactions in your body are not happening all the time. Only when you need certain reaction, that reaction is activated. So how do you activate a pathway? You activate by activating an enzyme. So if the enzyme is turned on, then it is active, so it will give you the reaction. If the enzyme is turned off, doesn't matter how much substrate you have. You may have a 10 times Km, it doesn't matter, the enzyme is not active, so it will not give you any reaction. And that is called regulation. A chemical catalyst cannot be regulated like that, whereas enzymes are highly regulated. They are either off or on. If they're off, doesn't matter how much substrate you have, there will be no reaction. Alright, so that is another reason why it is regulated. So what does that have to do with multiple steps? If you have multiple steps, that means you have these enzymes. So there are multiple opportunities for regulation. And as we will see later, all the reactions as I've written here are not like this. They are not going in one direction. They are reversible and irreversible reactions. And usually the irreversible reactions are regulated. So in a pathway having, let's say, 20 steps, maybe a couple of them will be irreversible, maybe 3, 4, 5 will be irreversible, and those are the ones that will be regulated. So more steps means more enzymes to be regulated. So more steps means more regulation sites. And that way the pathway can be controlled better. So in this one, if there are, let's say this is a pathway that has this branch and that is another branch coming into that pathway, we want to regulate this, not just this pathway that I've written here, this is also another pathway, you want to regulate both of them. You don't need both of those pathways all the time. Whichever is needed, that enzyme will be turned on and the pathway will be on. So if there are more steps, then you have more places where you can regulate it. Alright, so I'm going to erase all this. So there is a lot more to talk about regulation. So how does the regulation happen? The enzymes, as I said, are turned on or off, and there are basically about 4 different ways you can turn on and off an enzyme. So one is called feedback inhibition. So feedback inhibition means if you have a pathway like this, you can make some of these. I'm just drawing arbitrary pathways, so some of them will be reversible, some will be irreversible. Like this. So the ultimate goal of this pathway is to make this G, the product. But remember, once again, I mentioned last class that pathways don't have a beginning or end. The pathways are how we define it, that the goal is to make G, but for the cell, the goal is not to make G. It is mainly to continue further to make other things from G. All right, so for our purpose, we are looking at it as going from A to G, and we name that as a particular pathway. It has a name. And in that pathway, the purpose is to make G. Now, the cell must stop making G if it doesn't need anymore. Once it turned on the pathway, doesn't mean it will be on forever. It has to stop when enough of the final product has been made. So the final product, or any of the products in between, maybe G or F, will inhibit one of the earlier reactions in the pathway. So when enough of G has accumulated, you don't want the pathway to continue. So this will go and inhibit this enzyme. So let's say this is E1, this is E2, this is E3. So G will inhibit E2. And that inhibition will stop the whole pathway, because if you don't make C, then none of the remaining steps will happen. So this is called feedback inhibition, in which a product in a later step, it's not always the last step, because as I said, there is nothing called the last step in a pathway. So a later step in the pathway will inhibit an earlier step in the pathway to turn the pathway on, turn it off. And as we will see later, it is always actually an irreversible step. You won't find G inhibiting this enzyme, because this is a reversible one. It always inhibits an irreversible one. So that means nature has evolved to make this enzyme E2 in such a way that it has a binding site for G. That's called allosteric inhibition. Right? So feedback inhibition also works by the mechanism of allosteric inhibition, which, if you remember, means it binds to a site other than the active site where the reaction is taking place. So it binds to another site, changes the conformation of the enzyme so that it doesn't bind to the substrate. That's how it inhibits. So that is called feedback inhibition. You can also have a feed-forward activation. So when a pathway has been turned on, so let's say this pathway has been turned on by activating E2 by an activator. So some activator turns on the pathway, but then the cell has to be ready for the influx of C, D, E, F, and G. So it is getting ready for that. So there may be an enzyme that is activated. So normally, let's say this one, E3, this is E4, this is E5. So let's say E5 is usually present in a dormant state. It is not active. When this enzyme has been turned on, this C goes and activates this enzyme. So activation is written as a dotted line with an arrow with a plus sign, inhibition with a minus sign on the arrow. Minus sign with inhibits, plus is it activates. So this is called feed-forward activation. That means as you form the compound C, that activates a later step in the pathway, getting it ready for the pathway to come to G, because that's the goal of the pathway. So it activates a later step in the pathway which was normally dormant. It was inactivated by other means, and it needs to be activated, and that is done by one of the earlier products of the pathway. It activates a later step in that pathway. So that is feed-forward activation. Another method of regulation is covalent modification. Covalent modification means a covalent bond is formed. These are not allosteric inhibition is not forming a bond. It's just binding of two things. Binding of the activator or inhibitor to the enzyme. Covalent modification actually forms a bond. Yes.
Is FeedMore activation also allosteric?
Yes. Yes. So yes, this is also allosteric. That means it doesn't form a covalent bond. So whereas there are some methods of activation or inhibition that forms a covalent bond, and that usually happens in a process called signal transduction. Happens in signal transduction. For example, when you have a hormone in your blood and it goes to its target cell, the hormone doesn't really enter the cell. It sends a signal to have some end pathway activated or inactivated. For example, insulin. Insulin is too big to enter the cell. So it will bind to a receptor on the membrane. So here is your cell membrane. And let's say this is the insulin receptor. So this is insulin receptor. So I'm just giving the example of insulin, but it applies to all signals that go into a cell. So when the insulin molecule is secreted in the blood, so these insulin will bind to its receptor. And insulin doesn't really enter the cell. It's too big to enter through the membrane. So it binds to its receptor, and the receptor undergoes a conformational change and it activates a certain enzyme. So it will carry out a few reactions which I'm not going into. Let's say at the end it activates a protein. So there is an enzyme that will become active. How is it activated? So this enzyme, let's say this enzyme is like this. It's in inactive form normally. And it will activate the enzyme by putting a phosphate on it. So this enzyme will have a phosphate put on it. So once again, the P with the circle means it has 4 oxygens, PO4. So phosphate ion goes on one of the amino acids. Usually then it will be maybe one that has a OH group, because you need to form an ester linkage, or on an NH group, which forms a amide linkage. So any of the amino acid, not any, but one particular amino acid, this phosphate will bind and that becomes active. So there will be an enzyme. Every reaction in the body is catalyzed by an enzyme. So any enzyme that puts a phosphate on a protein is called a kinase. So this is a kinase enzyme that puts a phosphate. So kinase is not the whole name. So name of the enzyme will be whatever protein it is, followed by a kinase. Yes?
Does that say it says above receptor? What is that?
Insulin.
Oh, it's.
Or in any hormone. That's just an example. So insulin binds to the insulin receptor and it carries out a series of reactions, and at the end it activates an enzyme called a kinase that puts a phosphate on the protein that will carry out the response. So what is the response of insulin? Every hormone has a response. So the response of insulin is to bring in more glucose from the blood, to decrease the blood glucose concentration. So response of this final protein in the active form will be to take in more glucose into the cell. That's how you decrease the blood glucose concentration. Now, so this is called signal transduction. That means the protein doesn't enter the cell, but signal goes in, and then it works by forming a covalent bond on an enzyme and activating it. So that's regulation by forming a covalent bond. Now, once you activate this protein, it should not remain active forever. Right? It's not a one-time, once in a lifetime event. It's a every minute event, every hour event. So that means there must be a mechanism of inactivating it. And that's how you are responding to insulin whenever you have insulin or not have insulin. It's not just once reaction. So there must be a reaction that will remove this phosphate and convert it back to inactive form. So wherever there is a kinase, there will also be a phosphatase. So this enzyme is called a phosphatase. So phosphatase will remove that phosphate and make the enzyme inactive. So this is an example which I made up. You can put any hormone, any protein. This is an example where a protein is activated by adding a phosphate. There are other examples where protein is activated by removing a phosphate. So depending on, and that is always permanent, it's not at random. If it's a certain protein, if it is activated by kinase, then it will always be activated by that kinase. There may be another protein that is just the opposite. It can be deactivated by adding a phosphate. And usually the opposite enzymes, kinase and phosphatase, for example, if kinase is activated by adding a phosphate, then phosphatase is activated by removing a phosphate. It's just the opposite, because you don't want two opposite reactions to happen all the time at the same time. See, if one is active, the other must be inactive. So there must be another mechanism of removing or of activating the phosphatase, another mechanism for activating the kinase. So this is just an example where a protein is activated by forming a covalent bond or breaking a covalent bond. So that is called covalent modification. And as I said, usually if there are two opposite things happening, one of them will be activated by adding a phosphate, and the opposite one will be activated by removing a phosphate. Yes?
Is covalent modification only used to activate?
No, the deactivating, if something is activated by adding a phosphate, then it will be deactivated by removing that phosphate. That is also a covalent modification. You are breaking a covalent bond. That is also a modification. So forming a bond or breaking a bond, both are modifications. So that's one way that you regulate activity of enzymes, and not just one enzyme, it's a pathway, because this may be part of a pathway after this. So once this is active, then the whole pathway becomes active. Or if that is inactive, you turn the whole pathway off. You generally don't activate and inactivate every enzyme in a pathway. Cell does it very efficiently because pathways are coming, the steps are coming one after another. So if you inactivate one, then the whole pathway turns off. A fourth method of regulation is by regulating gene expression. Just today, the Nobel Prize for Physiology was announced. Anyone knows what it is for? No, it was for gene expression. So it is for regulation of gene expression, which means even anyone who has taken molecular biology, you know that enzymes are made from RNA, RNAs are made from DNA, and that is the gene. So if you have a gene here, if this is your DNA and there is a certain gene that is coding for a certain protein. So every gene will code for a protein, so even the kinase, phosphatase, they all are coded by some protein. So this gene is DNA, and when the DNA is turned on, this gene is turned on, it will convert to a RNA called mRNA, and based on the sequence of the mRNA, the protein will be made. Whatever that protein is, let's say it is the kinase or some other protein. So if the protein is not made, there is no question of making it active or inactive, right? So this is one way you make a pathway active, by making the protein. If the protein is not made, then there is no such question. So you regulate a pathway by making or not making a protein, and that process is called gene expression. So gene expression, it's a little confusing which step is actually gene expression. Is it the making of the RNA or making of the protein? And the answer is both. The purpose of making RNA is to make the protein. RNAs are not made at random and left like that. So if RNA is made, that means protein will also be made. So the two together is called gene expression. And in order to express the gene, right before the gene, in this region, there is a sequence of the DNA called the promoter. So every gene is preceded by a certain sequence called the promoter, and the promoter regulates whether that gene will be expressed or not. And how does it regulate? There is something that binds to the promoter. So let's say there is this molecule that binds to the promoter region. Promoter is this region of the DNA. So it binds to the promoter region and then it turns on the gene or turns off the gene. So if it turns off, then that is inhibiting. If it turns on, that is activating. So there are activators of a gene expression and inhibitors of gene expression. So this way then, these are the four methods by which a pathway can be turned on and off. Now there is a question of speed. When you want to regulate something, the question always arises at what rate is doing that. So which one do you think is the fastest of these processes? It will be just the binding. Binding is the first. Binding happens because two things are moving and they come together and they bind. That is the fastest process. And the rate of that is the rate of diffusion. Rate constant is the rate constant for diffusion, which is very fast. It's about 10 million per second. So the fastest is allosteric regulation. Both of these work by binding. So fastest. If you want to turn on or turn off a pathway, the fastest way to do it is by allosteric regulation. Covalent modification is actually a reaction. So every reaction takes a certain time. And so that will be the second fastest. And gene expression is the slowest because there are multiple steps involved. Something has to bind here, the gene has to convert to mRNA, mRNA will be then made into protein. And there are multiple steps. A protein has, let's say, 100 amino acids, so all those will be combined to make a protein, and each of them takes a long time. So making the final protein, and not only that, after the protein is made, there are more things to be done. The protein needs to be folded properly, because every protein has a three-dimensional structure. If it is not folded properly, then it is not active anymore. So the process doesn't end there. After the protein is made, it has to be folded properly, and then some proteins work by combining with other proteins. So that's the coordinate structure that we discussed before. So if it has to find the right subunit, if it is not the same one, then combined with that subunit to make the final enzyme. So that takes a long time. So gene expression is a long-term thing, so this is the slowest. So that means the allosteric inhibition works in seconds. So while pathway is turned on, at the very next second or minute, it may be turned off, right? For example, if there is an emergency and you're running, you don't want to keep running for an hour, you want to run for 1 minute, the process has to stop there. So that will happen this way. Alright, so I'm going to erase all this. So every pathway has a certain properties, and that applies to all pathways. So the first property is that all pathways are irreversible. So irreversible means, you studied in general chemistry, it means that they have a delta G negative. So if there are multiple steps in the pathway, you add all those delta G values of all the steps, and the end must be a negative number. All pathways will have a negative delta G, which means all pathways go in the forward direction. So if you have A, so if the pathway is on, then this whole process will continue to happen. If it is not happening, if it is not moving forward, then it is not a pathway, or the pathway has been turned off. If it is off, then it is completely off. If it is on, then it has to move forward. You have to make E from A. And that is also called a flux. Flux of a pathway is which direction the pathway is moving, and the flux of a pathway is always forward. So which means that no pathway will go in the opposite direction. If you are making pyruvate from glucose by the pathway called glycolysis, then glycolysis will never make glucose from pyruvate. So pyruvate can never go back to glucose using the same pathway. There may be another pathway that takes pyruvate to glucose, but that is a different pathway with a different name, different reactions. This pathway can only move in the forward direction. No pathway can go in the backward direction. So everything has a delta G negative, but in this, as I have shown here, this is a reversible step, this is an irreversible step. And these are technically called the metabolically irreversible step or metabolically irreversible step. Why is that adjective there? Because in a test tube, it may be completely different. Whether something is reversible or irreversible is decided by how much of the reactant and how much of the product you have, right? If A and B, if this is a reaction in a test tube, if B goes back to A and A goes to B, and it has the same rate, it has come to an equilibrium, then we call that an equilibrium reaction. The same reaction in a cell may not be at equilibrium, because in a cell you don't have isolated reactions like this. It is always part of a pathway. So A is coming from somewhere, and B is going to somewhere. And the cell always maintains a certain concentration of B. So in a test tube, if you have A and B only, it may be at equilibrium. In the cell, it may not be at equilibrium. So all these equilibrium and irreversible, these are relative terms. In the cell, it means under the conditions of the cell, under the conditions of concentrations of reaction and products, one may be irreversible and one may be reversible. But then, as I said, the flux is always moving forward, which also tells us that there is really no equilibrium step. And I mentioned this before, in a living cell, there is no equilibrium. No reaction is at equilibrium in the cell. Only time you reach equilibrium is when the cell dies. So that means all these steps that are written as reversible steps are not really reversible. This has a delta G that is slightly negative. So all reversible steps or metabolically reversible steps have a slightly negative delta G. Because remember, all pathways have to move forward. The flux is always forward. So you cannot really have a reversible step. You can have a close to reversible step, almost reversible step, which means they have a slightly negative delta G. And if it is a irreversible step, this is a highly negative delta G. Alright, so all reactions in all pathways will either have a slightly negative delta G or highly negative delta G. You cannot have a delta G equal to 0 because no reaction is at equilibrium. Even though the rate is written with an equilibrium sign, it is not really at equilibrium. Alright, that's one property of a pathway. Now, as I said, you cannot have a reaction going in a pathway going in the opposite direction, and that is easy to understand because if you have a reaction, so let me continue with this one. If you have a reaction that has a negative delta G, so if this is the reactant, then the product must have a lower delta G. So this is the amount of energy that is released. So there will always be energy that is released in a pathway. So A is going to B through these steps. Some of them may be reversible, some of them may be like this, and some going down. So if you have a reversible step that is slightly negative, if you have a irreversible step that is highly negative. But eventually it goes from A to B, which is at a lower energy level. Now, if you want to go from B to A, you cannot go in the reverse direction. Where will the energy come from? Right? You have to gain energy. And so, going in the reverse direction, you cannot use the same enzymes of this pathway. You must use different enzymes to go from B to A, because it will require energy. So, it needs ATP, and that means the substrate is different, and so the enzyme must be different. So that's why you need a different pathway to go back to make the original compound because of energy considerations. It is always a lower energy for the product, and so in order to go back, you will need to have ATP. And as we will see later, how much ATP do you need? Let's say if you have a pathway from A to B through multiple steps, and B went here, and this delta G is, let's say, about 30 kilojoules, 1 ATP, right? So you made 1 ATP in the pathway. If you want to go back to B, if you want to go back from B to A, you have to provide 30 kilojoules of energy. But that's not how it works. If you provide only 30 kilojoules of energy, that reaction will not be irreversible. All pathways have to be irreversible. In order to go back, you will need more than 30 kilojoules of energy. And so in order to go back from B to A, you have to provide extra ATP. So 1 ATP here, and in another step you put 1 more ATP. So in one direction, if you are making 1 ATP, if you want to go back in the reverse direction using a different pathway, you must provide more ATP than you actually got in the degradation pathway. So if you get 32 ATP by converting glucose to pyruvate, if you want to convert pyruvate to glucose, instead of 32, we will need more. We will need about 36 ATP. So you always need more energy to go back in the anabolic reaction compared to the catabolic reaction, because the reverse direction also must be irreversible. Alright, so people cannot see at the bottom of the board, so I will erase this one to write the second. Alright, so I'm going to erase this. Alright, people wanted a 5-minute break. We will do that after this second one. So all pathways have a first committed step, which means if you have A to, let's say, B, so you are going from A to X, and these are the steps. And there can be a branch. So let's say there is a branch. This one goes to another pathway. So not just one branch, there can be multiple branches in a pathway. Now, how do you make sure that X will be made? If this enzyme is turned on, let's say this enzyme is turned on, does it make sure that you will get X? That it doesn't, because this can go in this direction and go to the other product. So turning on and off this step or this step, as I said, the reversible steps are not really regulated, so this is an irreversible step. So if this one is regulated, so that this is turned on, if this reaction is turned on, it doesn't ensure that X will be made, because the pathway can go to C to D and then go in this direction instead of making X. So every pathway will have a first committed step, which means if that step happens, then the rest of the steps must happen to the end of the pathway. Which is not true for this one, but is true for this one. If this is turned on, then you will get X. Or if X is not the last one, so if this is X, then Y, and then Z. If this is turned on, you are sure that Z will be made. If this one is turned on, you are not sure that this one will be made. Sorry, or this one or that one will be made, because there are branches. So what is the first committed step? It is the first irreversible step after the last branch in that pathway. There may be other branches, so there may be a branch here coming in this direction, and one branch going in this direction. So which is the last branch here is this one, and the first irreversible step after that one, because irreversible steps are the ones that are regulated, not the reversible ones. So the first irreversible step after the last branch is the first committed step. So first committed step is this one, because if this is turned on, then the rest of the pathway will happen. So this way then every pathway has a first committed step, and as you can expect, where will the regulation happen? It will happen at the first committed step. Regulation will also happen somewhere here, because this also has to go to its own end of the product. So whichever is regulated here, whichever is irreversible, let's say this one is irreversible, then this can be regulated. So this will be the first irreversible step after branch. So this becomes the branch of this pathway. So after the last branch, this is the first irreversible step. So to regulate this product, you need to regulate this pathway. To regulate this product, you need to regulate this step in the pathway. So that's the first committed step of every pathway. So as people wanted, we will have a 5-minute break, and then we will continue. And anyway, in the meantime, if anyone has any questions.
There's 2 seats over
Everybody
there.
want to do some bellies? Deli?
He has to satisfy all the boundaries. And if there is any case, then you have a problem, like your disease. Because you want the final product X as well as the final product Y, but you don't have enough supply, then that will cause some problem.
Be able to talk.
That depends on how it is. That way,
And your card. Well, I tried
No, for me, I asked the question. You know, you don't use the reference. But I use the reference for every question except the last two. So when he saw me not using one, I put up the yes reference for all of them. I'm not
I was hit by a foot today. I was literally right over here.
I got hit by
It's so funny. The guy just walked out. Arabic. On camera. You can hear it. It's very clear. Crisp audio. No, I was watching the recording from last Monday's class. Because it's 2 hours, everybody got bored. They were just...
I signed up for it. The last one, the only one that's open. Yeah.
Unless when I go past
Big guys,
That's fine, I wanted to talk.
get a haircut.
Because you're considering that you can
where?
get the thing from.
We'll do some Arabic. Because that's starting
And then put after all our
Two haircuts like free haircuts. There's a haircut place next to the pharmacy. It's $100 per hair. What? That's wild. Because apparently the guy, Jay, there's a stylist or whatever, and he does a lot of stuff. He puts the black gloves on. Yeah, that's actually good.
what do we...
That's all. رحت الجو خلاص ولا you already went to the gym?
I just didn't write it.
I'm going afterwards. I'm gonna hit legs today. Can we come? You squatting 225 today. Huh?
said, I don't know about squat. Also, what happened? Why are they all sick? Bro, first, I'm convinced Roman got me sick. He can say he was asymptomatic, whatever he is.
I'm also convinced Roman got me sick.
I got sick, not even a day later he gets sick.
Why am I coming with a bunch?
Yeah.
You want anything from the coffee shop?
Thank you.
Croissant, bagel, donut.
I'm paranoid because I dropped half of orientation. One person, those are like
Like, you just like balance out.
life is perfect. So like, you don't want to raise the ISO on the camera too much because it gets grinding. The picture becomes unusable. So one of the lights, she that girl, she walked
Like,
like
you remember? I mean, like,
I never forget a place that dropped my lights. Like she walked right into the tripod, the whole thing just fell forward, and the battery jumped in the ground. And I was like, oh, it's broken, guaranteed. And then I also shoot with my friend.
Was it?
No, it was fine. I shot with my friend like 3 months ago. One of the older lights he took because it was a birthday party. And there was a pool there. There was kids. So a kid ran face forward into the light goes into the pool, like fries all the electronics. It took me like $150 to fix. Which is, I got it fixed and I got it fixed. Because they paid, the clients paid for it because it was their kid. It was supposed to be a kid at the party. So they're like, okay, we'll get you a new one. But then I was like, I can get it fixed for cheap. It's not bad. The light costs $500. I'll just get it fixed. But it was good enough. And then he dropped one of my other lights. The one that goes right on the camera. Smashed the light to in half. But it's okay.
Yeah, I remember once I was at a football game. My favorite guys were like, and you know, they have their own and they go like, people, and I was always there. And I was like, oh my god, I'm like, I'm trying to get a good drop. And they hit the ball. Yeah, and they literally hit it. But I think I never paid for it. I never paid for any of my
No, I know, bro. Like, it was the school's white lenses. Like,
yeah.
It was like, what, a 400?
It was a 400. It was a 400.
2.8?
Yeah. It was a 400. Yeah. That's about it. Really like the whole thing. And then I didn't pay for it, but like, you know, I was.
They scratched it.
They like scratched the whole thing. And I had it on big because I was
And I've seen the biggest ride because Derrick Henry had like a 50-yard slide and so the one thing, and he said he had the answers on
What is this?
Yo, you can't be drinking
Let's
that.
not do that. Well, no, that's not healthy.
He goes, I have a headache.
No. Let me feel the can. It's gonna make it worse. You can have like that's all you're like adverse effect or something. I'm serious. I'm helping as your friend. You're not drinking all that. You feel better this way? No. That's not.
They have to be. Bro, they make it that size on purpose.
No.
It's not like a human Tesla.
It's like what? That's light work. How much coffee?
This is his 7th one today.
Is this like one serving
He
size?
drank 2 already. Who is my shirt?
So the third property of a pathway is that some pathways may be compartmentalized, which means they are happening in an isolated region, independent of what is happening somewhere else. For example, if you have, they can be compartmentalized at the organ level. So one organ may be doing a reaction and another organ can be doing a just opposite reaction at the same time. So you may be doing glycolysis in one type of cell, and at the same time another type of cell, maybe in a different organ, can be doing just the opposite, making glucose from pyruvate. So that is all allowed. And that always happens also. So because they are happening in different places, especially in eukaryotic cells or organisms, they can be happening in different parts of the body and doing opposite reaction. Within the same cell there are organelles, and the organelles may be doing different reaction. For example, fatty acid synthesis will happen in the cytosol, and fatty acid degradation will be happening in the mitochondria. It usually doesn't happen at the same time because they are regulated. If one is on, the other is off. But there is no reason why they cannot happen because they are in different places. They are in different compartments of the cell. Compartmentalization also happens at even a lower level, even at a molecular level. For example, a protein is large, and one part of the protein may be doing something, another part of the same protein may be doing a completely different reaction because they are compartmentalized. And also because they are compartmentalized, sometimes it helps the reaction to increase the rate. For example, if this is at the molecular level, if this is a protein, carrying out, let's say, more than one step, like carrying out 3-4 steps. So here you have A going to B, and this part of the protein carries out the reaction B to C. If these were two separate proteins, what would happen if these are two separate proteins? A to B happens here, then B has to leave, and then it goes into the cytoplasm and gets diluted. So once B is formed and it leaves, it becomes diluted, and then this has to be captured by the second enzyme, which won't happen unless you reach a certain concentration of B. So that makes it inefficient because they are separated. But if they are together, then this B directly goes from this one to this one without getting diluted into the medium. And there are many examples of this kind. So these are multi-subunit enzymes. Many enzymes work like this. They are 4 or 5 different enzymes are all together. And the whole purpose of these multi-enzyme complexes is that the intermediate doesn't leave, because the intermediate is formed in small amount. If it doesn't leave, it is basically channeled from one subunit to another, then the concentration of B for the second one is high and the second reaction happens immediately. And that's a common reason why many proteins are present as protein complexes rather than one separate enzyme. And we will see many such examples where you have large complex, and in all cases the reason is basically channeling of the metabolite. So this process is called metabolite channeling. Where product of one reaction is directly transferred to the second enzyme without releasing it into the cytoplasm. And that way the concentration of the second substrate remains high in the enzyme. So those are all different examples of compartmentalization, which means two opposite reactions can be happening at the same time in different places, or the same reaction may take multiple complexes, multiple enzymes to happen, and all those enzymes may be together in one location rather than multiple locations. All right, but that's not true for all pathways, so some of them, so that's why it says may be compartmentalized, so it doesn't have to be. But one thing that happens is all pathways are regulated. I've been mentioning this many times. It is always regulated at the irreversible steps. And why is that? Why do you want to regulate the pathway at the irreversible step? And that this will be the last thing on the metabolic pathways in general. After that, we will move on to specific pathways and start with the carbohydrates. So we'll move on to the chapter on carbohydrates. So why is it regulated at the irreversible step? So regulation means turning on and off, like turning on and off a switch. So if you enter a room and you turn on the light, you expect the light to turn on right away, within that second. You don't want to wait for 1 minute for the light to turn on. So in the same way, if you regulate a pathway by turning it on, you want to turn on immediately and not after some time. In the same way, if you turn off a switch, you want the light to go away right away. So if you want to turn off a pathway, you want the effect to happen immediately. So let's look at two situations. One, regulation of an irreversible step. Or regulation of a reversible step. So then let's say there is a reversible step before this. So if you have a reversible step, and you want to regulate this one. If you want to regulate the reversible step, and there are two situations, let's say we are doing with turning on. And then we will talk about turning off. So if you are turning on a pathway, that means this pathway is off. It's normally off and you are turning it on with some regulator. So the regulator binds to this, and this pathway is turned off, which means there is no B present. Right? The pathway is off, so reactions are not happening, so there is no B present. That means the concentration of B is 0. When will this second step happen? This is an irreversible step. What is the definition of an irreversible step? Definition of an irreversible step is that this concentration is high and this concentration is low. That's how all irreversible steps are. You have high product reactant, low product concentration. That's what makes it irreversible. And if it is an equilibrium, then they are about similar amounts. Not same amount, but similar amount depending on the equilibrium constant. So if this is an equilibrium step, that means you need a significant amount of B to start this second step, because this is an irreversible step. B must be high, C must be low. So B concentration has to reach to a high level before C can be made. But B, you are starting with 0. This is already turned off and you are turning it on. So you turn on this one and then you wait for B to go up. It has to go up to a certain level, which is defined as a high level for this reaction, and then only C can happen, then only the rest of the pathway can happen. So activating or turning on an irreversible step is not desirable because the cell has to wait for B to go up so that the rest of the pathway can happen. On the other hand, if you are regulating an irreversible step, so even if there are reversible steps after this, the definition of an irreversible step is that this is already low compared to A. And the second step will start at a low concentration of B, because that's how the pathway has been made by the cell. Whether it is reversible or irreversible doesn't matter. The pathway has been made such that this reaction will happen even at a low concentration of B. So when you turn it on, as soon as you turn it on, small amount of B is made and then the pathway starts. Here you need a large amount of B for the rest of the pathway to happen. And that makes sense to regulate an irreversible step, because definition of an irreversible step is that B will be low and still the rest of the pathway will continue. Whereas for a reversible step, B must be high and then the rest of the pathway will continue. So the cell has to wait. So it's like turning on a switch and then waiting. So that's not desirable, so that's why this is regulated here. Now, if we are turning off, same thing. Turning off means the pathway is already on. And let's say you are going to 70. This is also going to 60. So if you are regulating this step by turning it off, then when you turn something off, you want to turn off immediately. But if you turn off this enzyme, there is always a large amount of B, because this is an equilibrium step. So B concentration is very high, similar to A. So if you turn off this enzyme, there will always be some B left, and all that has to get utilized and go all the way to the end for B to come down, and then the pathway stops. Alright, so that's why you don't regulate an irreversible step. You don't turn off a reversible step, because the product of that step is already high, and all that needs to be consumed in order for the pathway to be turned off. So that will take time. Whereas if you are regulating this one, the concentration of B is low, and as soon as you turn this off, this goes down to zero right away. Because this is already zero, already close to zero, and you turn it off, it goes down to zero, and the pathway stops right away. So that explains why pathways are always regulated at the irreversible steps. And if there are several irreversible steps in a pathway, then maybe all of them are regulated, may not be, it depends on the pathway. But usually the first committed step will always be a regulated step, because that decides which pathway will be turned on and off. So first committed step is always regulated. Besides that, there may be other irreversible steps in the pathway which may or may not be regulated. Usually they are, because that's how nature has evolved these pathways to regulate at each irreversible step. Alright, so as I said, that's the last thing about generalized information on pathways. We will talk about specific pathways later. First, introduction to carbohydrates. Any questions up to this point? And that actually puts us slightly ahead in our schedule, because we need that, as I mentioned before, the carbohydrates, and the pathway related to carbohydrate takes more time than what is allotted. So we will need this extra time, as well as we will need extra time from future chapters, like the nucleotides. Doesn't require so much time, but the carbohydrates will require more time. So as the name suggests, carbohydrate is carbon and water. So that's the general formula of carbohydrate, carbon C and H2O water. So if we combine that, that is the formula of carbohydrate. Carbohydrates can be aldoses, or ketosis. So that means aldehydes or ketones. So the simplest carbohydrate will be a 3-carbon. So aldehyde means you have a CHO. So this is a 3-carbon aldehyde, and that is called glyceraldehyde. 3-carbon, remember 3-carbon OH is glycerol. So 3-carbon with an aldehyde group will be glyceraldehyde, 3-carbon with a carboxylic group will be glycerate or glyceric acid. So this is glyceraldehyde. And all carbohydrates have lots of OH, and this also has OH, one OH here and one OH is on this carbon. And this is a chiral carbon because one will be a hydrogen, the other will be a OH, which means you will have 2 enantiomers of this. Enantiomers in organic chemistry you studied are R and S. Biologists usually use R and S, so they use the D and L system, which is based on the Fisher projection. And for the DL system, you have to have a rule about how you draw the Fisher projection. Every group, every new type of compound must have its own rule. Like amino acids, we had its rule, and for carbohydrate, the rule is that when you draw the structure, the aldehyde must be on the top. Remember, if you have the aldehyde at the bottom, then the whole thing changes. You cannot do that. So, according to rules, the aldehyde must be on top, and when you do that, if the OH is on the right side, then that's a D sugar. So the first chiral center from the bottom, when you write with the carbonyl group on top, the first chiral center from the bottom decides whether it is D or L. This has one chiral center only. For the other carbohydrates, there will be more than one chiral center. But the first one from the bottom decides whether it is D or L. So this is the first one from the bottom. There is only one in this, and this is on the right side, so it is D-glyceraldehyde. And if it was on the left side, like this, then this would be L-glyceraldehyde. So this way there can be many carbohydrates. This is a 3-carbon carbohydrate. You can have 4-carbon, 5-carbon, 6-carbons. That will increase the number of stereoisomers. Half of those stereoisomers will be D and half will be L. And the reason the biologists love this kind of system is because nature makes only the D glucoses or D sugars, and so 50% of the sugars don't even exist in nature, so we won't talk about that, and those are the L sugars which don't exist. So all sugars are D sugars. So if a certain sugar has 4 chiral centers, then it should have 2 to the power 4, which is 16 stereoisomers, of which 8 will be D and 8 will be L, and we don't even bother about the L's, so we'll talk about only 8. Like glucose has 4 chiral centers, so there will be 8 possible stereoisomers that you can find in nature. You can, but do you actually find them? And the answer is no, you don't find all those, or even if you do, it may be very insignificant amount. So they are present in variable amounts. And so most of the stereoisomers that are possible, we won't even talk about them because they are not very significant. So we will talk about only basically a few of the sugars which are the major ones present. So 3-carbon sugar is important. This is a glyceraldehyde which is present in many places, in many pathways you will find this. 4-carbon, not so important. 5-carbon is important. Why is it 5-carbon important? Which sugar has 5 carbons? Ribose, and that is present in DNA and RNA, deoxyribose and ribose. So 5-carbon is important. So if there are 5 carbons, then this last one is not chiral. The first one is not chiral. So these are the 3 chiral centers. So that means there will be 2 to the power 3 or 8 stereoisomers, of which 4 will be D. And doesn't mean all 4 of them are present equally. The only thing that you need to know is the ribose. The other ones are not so important. So there are only a few sugars that you need to know the structure of. And ribose is also easy to know because all the OH groups are on the right side. So right, right, right. So this is D-ribose, very easy to remember, all the OH on the right side. And because only D is present in nature, most of the time people don't write the D. So ribose means D-ribose. And then of the 6 carbon sugars, you will have one more chiral center. So there will be 16 stereoisomers, of which 8 are D, and all 8 of them you don't need to know, they are not present in significant amounts. So which one is the major sugar that is present in nature? Is the 6 carbon sugar is glucose. So that structure you need to know. And the easiest way to know that structure is where the OHs are. So these are right, right, left, right. So if you remember that sequence, right, right, left, right from the bottom, that is glucose, because if you remember glucose, the other ones will follow from this. So these are the only 2 structures you need to basically memorize. Ribose is easy, all 3 are on the right. Glucose is right, right, left, right. Right, that's easy?
Yeah.
Alright, so OH, OH, OH. So this is glucose. And I'm not writing the D because all of them are D, you don't have the L. And that is decided by this OH. Once again, the first chiral center from the bottom decides whether it is D or L. So if this was on the left side, that would be L. So which other 6 carbon sugar is somewhat common besides glucose? Yes, fructose is not this one, there's not an aldose. Anyone know sucrose is not, sucrose is a disaccharide. There are 2 other sugars that are present in relatively significant amount, and one of them is galactose and another one is mannose. So mannose and galactose are the other 2, and if you remember those as how is it different from glucose? So 2 things to remember. Mannose is different at C2. Galactose is different at C4. Mannose at C2, galactose at C4, the rest of the structure is the same. So if you want to draw mannose, you draw exactly the same way as glucose, but at C2 it will be opposite. So you have CHO. So right, right, left, and instead of right, this is left. So it is different at this carbon from glucose, and that is mannose. So mannose is present in small amounts, but still significant. And another one is galactose, and that is different at C4. So once again, you draw the 6 carbons, CH2OH at the bottom, and then C4 will be this one. So 1, 2, 3, 4. This carbon will be in the opposite direction. So you have OH. This one is to the left. So follow the glucose structure at C4, you make it the opposite. So that is galactose. And where do you find galactose? You said it's important, it's present. Where is it present? Galactose is present in milk sugar. Lactose is a disaccharide of glucose and galactose. Right, so that's the sugar that gives many people the problem of digestion, because many people don't have the enzyme to digest lactose, which is a disaccharide of glucose and galactose. Alright, so besides these, you also have the ketoses, and a ketone will have a keto group instead of aldehyde group, right? And obviously you cannot have a ketone at carbon 1, right? Why not? Can I have a ketone at carbon 1? No, why not? It has to be between two carbons, so it's very obvious that you cannot have a ketone at carbon 1, so the ketone has to be at carbon 2. So if you have a ketone in this position, so this will be so this will be CH2OH, this carbon, and this will be the keto group, and then this will be CH2OH. So CH2OH, CO, CH2OH. So this is the corresponding ketose of the 3-carbon sugar. And how many chiral centers does it have? None. So ketoses will always have one chiral center less than the corresponding aldose. Why? Because carbon 1 obviously is not chiral, because it's CH2OH, and this carbon 2 is now a 3 bond, not 4 bond, so that is also not chiral. So if this has 1 chiral center, this will have 0. Ribose has 3 chiral centers, so the corresponding ketose will have only 2. So there will be 4 stereoisomers, 2 of which are present in nature. And for 6 carbon sugars, there are 4 chiral centers in the aldoses, so in the ketoses there will be only 3, so that will mean 8 stereoisomers, of which 4 are present in nature. So always 1 chiral center less for the ketose. So what will be the name of this ketose? 3 carbon ketose, what will be the name of this one? This is a very important one, always present in the cell. This is derived from acetone. If you remember the structure of acetone, so this is acetone, and it has 2 hydroxy group on the acetone. So the name of this one is dihydroxyacetone. Dihydroxyacetone, which is an important intermediate in glycolysis. Glyceraldehyde is also an important intermediate in many pathways. So in the same way, ribose will have a corresponding ketose called the ribulose, which we are not studying because ribulose is important in what? Anyone knows what is ribulose? Where do you find ribulose? That means ribose with a keto group here instead of the OH group. That is called ribulose, and ribulose is important in photosynthesis. So that's where photosynthesis starts. And the first step of photosynthesis is started with the ribulose. That means the sunlight is captured, and the first product that is formed, the substrate for that is ribulose. Of the Calvin cycle, if anyone has studied in biology, ribulose is the first substrate for that. But we are not studying photosynthesis in pharmacy, so it's not important for us. So what is important is the corresponding ketose of glucose, and what is that one? Everyone knows this, right? What is the corresponding ketose of glucose? It is fructose. So to draw fructose, then you will just put a keto group here. So this will be then CH2OH, and then keto group. So this is fructose, and once again it is D-fructose. All sugars are D. That is based on this CH2, so on this OH, the first OH from the bottom, first chiral center from the bottom. So this is the corresponding ketose of glucose, and that's the only one you need to know. You don't need to know any other ketose. So fructose is the most common one. So basically these are the sugar structures that you need to know. Of which glyceraldehyde is easy because there is one chiral center, obviously it is on the right. Ribose is easy because all three OHs are on the right. And glucose, the only one you need to remember is right, left, right. Fructose will be the corresponding keto group at 2 positions. Mannose and galactose are present in very small amounts in different places. They differ at C2 and galactose at C4. Alright, so I'm going to erase this. And keep just the glucose. One thing that we didn't do from the energy part of it, which we will do when we come to glycolysis, is about the high energy compound ATP. Why ATP is always used. Right? I keep mentioning that whenever you need energy, we use ATP. Why ATP? Why not something else? So that's a topic that we will cover when we do get into energy utilization in glycolysis. So for carbohydrates, this is the glucose structure which has an aldehyde, and if you studied in organic chemistry, if you have an aldehyde, that can react with the alcohol. So if you have, the alcohol can be water also. So if you have an alcohol, ROH, so this reacts here, and you get this product. And that's an equilibrium reaction. And it happens by itself. You don't need to heat it, you don't need to do anything, it doesn't need a catalyst. It's just when you dissolve any aldehyde in water and there's an alcohol, they will form this product. And that is called a hemiacetal. It's called a hemiacetal because this also has a OH group which can further react with another alcohol. So if you react this further with another alcohol, let's say R prime OH, then you form a ether linkage here. This chain is here, so I'm forgetting the chain. Alright, so this is called the acetal. The first reaction happens by itself, no need for any energy, no need for anything, it's just an equilibrium reaction. The second reaction is difficult, you need a high temperature to do this. And it also means that the second one is irreversible reaction, it goes in one direction only. This is not a biological thing, it's an organic reaction in a test tube. But as soon as you dissolve any aldehyde in water and some alcohol, it will give you this. It's an equilibrium reaction, you don't have to do anything. And then that can react with more alcohol only under extreme conditions. That means you actually have to do a reaction, you have to do a reflux at high temperature and a catalyst. You need acid catalyst. So difficult reaction to do, but you can do that. That means you can add the second alcohol molecule also, then it will form an acetal. So if this second alcohol is also part of the chain, so if you have an aldehyde, and here is your chain, and that has the OH, it is the same as this one. You get a ROH reacting with this aldehyde group. So within the same molecule, the alcohol can react here. And then you have a ring with because one of the OH groups in the same molecule is reacting at this, so that forms a ring structure. And this situation is there in all carbohydrates because carbohydrates have lots of OH. And so this OH can react at this carbon. And once again, you don't need anything, it will just happen by itself. So as soon as you take some glucose and dissolve in water, this OH will react here and give you a ring structure. Now, before I draw that ring, let's look at this compound here. This is an aldehyde, this is achiral. When the alcohol reacts here, this becomes a chiral center. So you have created a chiral center. So that will give you two isomers, right? Because this is a chiral center, there are two possible stereoisomers. So basically every sugar, when you are taking glucose or any other sugar, when it forms a ring, it actually gives you two rings. And the structure of that is like this. This is the ring. This is the oxygen. This oxygen forming a bond with this carbon. So you have a ring from oxygen to carbon, that is this oxygen. And so this carbon is carbon 1, so this is the carbon 1. So 1, 2, 3, 4, 5, and the sixth carbon, which is this one, is like this. And the other OH groups here will be this way. It's easy to remember again glucose. This is called the Haworth projection formula. So you will find sugars in the book written drawn like this. And for glucose, there's only one that we need to know about that is easy to remember. This CH2OH is up, next one is down, next one is up, next one is down. And this is a new chiral center created. Remember, when the alcohol reacts at this carbonyl group, you have created a new chiral center at this aldehyde position. And that is this carbon 1. So this carbon 1 then will have the OH group either up or down. Those will be the two possible isomers that will form when it forms a ring. So if the OH group is pointing up and the hydrogen down, or the OH can be pointing down and the hydrogen up. So whenever a linear carbohydrate forms a ring, there will be two structures formed, and they are both glucoses because it came from glucose. It's an equilibrium reaction. It cannot have a different name, but we have to differentiate the two isomers. So one is called an alpha isomer, the other is a beta isomer. The beta is the one that has the OH on top. So this is beta glucose. This is alpha glucose. So alpha has the first OH at carbon 1 pointing down, beta has the OH at carbon 1 pointing up. Yes, yes.
So carbon 1 is the chiral center?
It's a new chiral center, yes. Once again, look at this reaction here. This is the aldehyde carbon, that's where you have created the new chiral center. And so carbon 1 is the one that will give you the two different isomers. One is the alpha isomer, one is the beta isomer. And it came from the same glucose. That means glucose always exists as a mixture of two isomers in solution. And that is true for all the carbohydrates. All the aldehydes exist as a mixture of alpha and beta. And these are called anomers. So this is another term used. So anomers are the two stereoisomers at carbon 1, or the carbonyl carbon. This was the carbonyl carbon, and now it is a chiral center. You get two isomers because of that, and those are called anomers. Or you can say anomers differ from each other at the anomeric center, or anomeric carbon, which is the carbon 1 in this case. And how do you recognize the anomeric carbon given a structure like this? And we will look at other structures. It's easy to figure out which carbon is the anomeric carbon. And that is the only one that is bonded to two oxygens. This is the only carbon bonded to this oxygen and this oxygen. There is no other carbon bonded to two oxygens. So anomeric carbon, even if you are given a structure which you have never seen, it's easy to spot which is the anomeric carbon. The carbon that is bonded to two oxygens is the anomeric carbon. And why is that name important? Because many reactions happen there. Most of the reactions you see about carbohydrates happen at that carbon. If you form a disaccharide, it is happening at that carbon. If you are forming a acetal, that means this next step, this alcohol reacts with this alcohol to give you the ether, but there are so many alcohols in carbohydrates. Which one does it react with? In other words, if you take glucose and do this reaction with any alcohol at whatever conditions are needed, out of so many OH groups, which is the one that will form the ether linkage to the alcohol, it will be the carbon 1. The anomeric carbon is the most reactive carbon in glucose, and that is the one that reacts. That is also called the reducing carbon because it is very easily oxidized. So if you oxidize glucose, this is the carbon that gets oxidized and the other carbons are not. So all the glucose tests that you do, whether you do it in the lab yourself or someone else does it for you in an analysis of blood glucose, for example, they will be looking at the anomeric carbon because that is the reactive carbon. That is the reducing center, which means it easily gets oxidized. Alright, so every time it is the C1 that is important because that is the reducing OH, and any sugar that has a anomeric carbon, that means it has the carbon attached to two oxygens, that is also called a reducing sugar because this is the OH group that is the reducing pocket. This gets easily oxidized, and so all the tests of glucose depends on that OH group. So coming back to this reaction, as I said, you can do a reaction of this one. So if you take this mixture and react it with, let's say, methanol, so that will form an ether linkage. I'm not drawing the rest of the structure. Or the beta, this is the beta one will give you this ether, and the alpha one will give you this. And they are not in equilibrium with each other. They don't interconvert. Once you form the acetal, they don't go back and forth. So this is a irreversible reaction, this is a reversible reaction, so they go back and forth to the linear form. So the ring structures can convert to the linear form again converts to the alpha and beta. But if you lock this with another metal group, then that will give you either the alpha or the beta. So this is the alpha, this is the beta, and they don't need to convert. So this will be called the beta metal leukocyte, and this is alpha metal leukocyte. So this way you can form the glucoside. The general name for all carbohydrates is a glycoside. Glycoside means any of the carbohydrates reacting with the methanol. Glucoside means glucose reacting with the methanol. Alright, so these terminologies you will see frequently. GLY applies to all carbohydrates. GLU is applied only. Alright, yes, that's we'll stop here.
Is the methyl glucoside a result of like that reaction that you drew there with?
Yes, this is the same reaction. This is the reaction with any alcohol, so this is the same reaction. That means this reaction doesn't go back. You need to do it at high temperature and at this, and it doesn't go back. So this doesn't convert to those. Alright, so we will stop here. You will have a next quiz maybe in 2 weeks from this Tuesday. That will be 40 hours.